Carmen, who is going to talk to us today. And I'll let him introduce himself.
Thank you. Okay, I'll try to make it a lightning talk, so I'll try to be quick and yet hopefully cast a bit of light on some interesting problems. So I'm from Brunel University, and this talk will be about not only connecting data, but really connecting knowledge into larger coherent models. And this talk is about a project we are conducting also in Brunel as part of a bigger consortium for the research program funded by DARPA. The program is called Big Mechanism, and it's about extracting large mechanistic models from scientific literature about certain domains. And so this is quite a lot a use case talk. So I'll be telling you how we use linked data and probabilistic techniques to address our task. And as much as it is a use case also to present here, this whole project was an exercise for me. I come from a bit different corner. My background is mostly in the more foundational knowledge representation and reasoning, so I will also focus more on the more core knowledge representation and reasoning problems. All right, so where does the problem start? So scientific community has a big issue with the publishing deluge. There's a lot of papers, so scientists are the strange creatures that solve the cutting-edge technological problems using sometimes not so cutting-edge techniques, so using chalk and blackboard. And also the publication outlet still traditionally goes into journal papers or conference papers expressed in a natural language, in text, and the whole knowledge that is delivered is really encapsulated in this form, which causes obviously a lot of problems in accessing this knowledge, in combining it, and making the best use of it. So just to look at a few numbers, since 17th century, where the proper empirical science started, there have been around 50 million papers published. Just last year, there have been 2.5 million papers published in this traditional form. And it's not obviously a constant factor, because each year there's more and more of these papers still being produced. So some estimates vary a lot, obviously, but some of them says that the output is doubling every 9 years. So we can easily expect we're gonna drown in these papers very soon. And there are, of course, a number of these negative effects. So one of them for the whole science is that science narrows down, in a sense that no one is able to read these papers, and we tend to cite more and more a narrow pool of leading papers. So there is a huge tail actually of work and results that are never even accessed and used for further research. There is a fragmentation of expertise. So simply speaking, nobody understands the big picture anymore. This project is focused in this stage on cancer biology domain, and one of the bold statements motivating this research is that perhaps the cure for cancer is already there. You just have to connect the dots, connect the fragments. So what can we do about it? Obviously, the whole philosophy of publish or perish also leads to negative effects for the quality of results. It turns out that lots of experimental results reported in biomedical sciences are actually non-reproducible. Again, there are different estimates, but some studies have shown that actually 50% of results published in biomedical domain are non-reproducible. And that's a dramatic thing for science, because we're basically building a huge structure on water. We don't know how it really stands, and we should know. So the goal of the Big Mechanism program funded by DARPA is to develop novel AI technology for extracting large mechanistic causal models, explanatory models from the literature, and express them in formats that can be accessible and processable by computational agents. Because obviously more and more tasks can be done with computationally intensive techniques, and if we manage to employ computational agents to do analytics, to do predictive reasoning, basically we can automate a lot of science, a tedious part of science. Of course, we never replace scientists at his position, but we can benefit a lot if we scale the whole process. So in case of our cancer biology domain, the goal is to automate the three-stage process: reading, assembly, where we assemble those mechanistic models. In this case, there are so-called activation pathways. I don't know how many people know a lot or enough about biology in this room. I'm not one of them, for sure, so excuse me if I'm talking a bit not a very correct things about biology, but I'll be using just a few examples there to motivate it. Yet the goal of the project, the overarching goal, is to develop technology that can be easily transferred to other domains, because the same problem actually persists in many other domains. So our part of the work which we do in Brunel is focused on the assembly process. Now, what are the challenges? Well, if we take a research paper and we look at the sentences that are of interest to us for building such models, we can see we have a lot of issues of extracting the actual knowledge from them. So some of them come from the use of natural language, and they are well known, especially in any text mining domain. So there's ambiguity, vagueness, and modality of natural language. Sometimes authors say, well, we strongly conjecture this result holds, which is very different from saying we know this result holds or we've proven this result holds. There is a general quality and reliability issues regarding the sources. We have number of journals. Some of them are not worth much. Some of them we consider very important and very valuable. There is obviously an inaccuracy of extraction pipeline. There are the typical V's of the big data, so the volume, the variety, volatility, velocity. So it's not only about, well, picking out all these sentences from the papers, but actually managing the whole amount of them that come out this way. Finally, there are bigger problems, deeper, more fundamental for biology especially. There are inconsistent, inconclusive, or non-reproducible results reported in this paper. So we have to think every time whether the sentence that we picked from the paper, does it really say something that's of biological validity or not? There are contextual assumptions, omissions, gaps, so things that we as human readers and human experts that work with this domain, they can kind of smoothly overcome, but they become a real issue once you want to formalize it and build a coherent model in automated fashion. So knowledge assembly is a process of reconstructing complex knowledge from these contextually asserted atomic statements and data fragments. And traditionally this pipeline would look something like this. So basically we extract knowledge, we collect all these sentences. So for instance, a protein A can associate with protein B. It's just one finding in a paper. Then we do the whole clearing and reconciliation of these facts and filtering and aggregating them. And then we come up with knowledge, what has been found in the paper, in all this literature. And at the end, the last stage, we actually form this biological model, so this activation pathway. But this model of knowledge assemblies is not the best. It's actually it has issues, and we want to, within this project, we want to address these issues by introducing a certain new paradigm of assembling knowledge. Basically, in this framework, we want to consider all contextual information and all the evidence-related information extracted from papers as part of our knowledge base, so that we can close the entire cycle of knowledge assembly in a constant update assembly loop. Whenever we get new pieces of evidence, whenever new information is extracted, we dynamically should be able to update what we know, because our knowledge changes dynamically. And only then we can really ask the right questions to an expert, because expert input is always very expensive. Whether it's an experiment or a human annotator, it's a very expensive thing to ask. So we need to ask right questions. And only then we can decide whether the knowledge we found in what way it contributes to the final model. Does it corroborate the model that we have already? Does it stay in a conflict with it? There is never an ultimate model. The way the science works is by constant updates and evolution of our domain models. So in this picture, the evidence is again the sentences that we extract from papers, like A can associate with B with certain extraction accuracy published in certain journals. So we take all this contextual and additional uncertainty information to include it in the knowledge base. Then, given all this evidence, we can answer a question whether certain events, so in cancer biology we deal with what we call events, so binding of a protein A by protein B. This is an event, a molecular interaction that will later on form a bigger structure. We can ask the expert, we can run experiments, and we can finally see to what extent the model is corroborated or remains in conflict with our findings. So what kind of linked data resources do we use to achieve this? So in the first step, we want to build a huge knowledge graph of all this information and connect the dots as we should. So basically we use a number of ontologies. These are biomedical ontologies, gene ontology, biopacks, molecular interactions, and we use, so this is a domain level knowledge, but also then we use the whole meta-domain, meta-level knowledge, which is concerned with uncertainties, so all type of information about how probable statements are in terms of different categories of uncertainty. So whether what's the extraction accuracy, what's the textual modality involved there, what's the provenance, confidence, all these types of uncertainty and are really important, and the assumption is we shouldn't filter them out and compile them out too early, because later on, once we we get more evidence, we should be able to reconsider our previous decisions. So we have this information about uncertainty, about different qualifications of the information collected, Provenance Document. Further, we use a number of data sources regarding, for instance, journals and bioinformatics databases. And obviously we use identifiers, particularly for biochemical entities, also for journals, articles. There's more to it. This is just a short version. Identifiers are actually very important here, and I will explain why in the next slide. So this is the ontology. Of course, it's nice for listing all the concepts that are relevant in our domain, but how is the data really connected? Well, it more or less corresponds to this model, a two-level model in which on one level, the evidence-related level, we collect all information about findings from the text. So the basic entity here is a statement. One statement is one finding digged out by the text miners. And we know a lot of things about this statement, right? So we know it has some textual expression in this article. It has certain truth value. That's the important thing, because two different statements might speak about the same interaction, but express different truth value. One says the interaction holds in the given context, another one says it doesn't. So all this information is collected and connected as here. Then on the second level, on the domain level or knowledge level, we actually represent what the statements are about. So really we formulate and express this structure of these events that are relevant. So the basic entity here is event or a molecular interaction. It has certain type. There is a number of these types more or less settled in the community and in science. And it has certain participants, and these participants are also explicitly represented there. Now let me show you just a small example of how this is kind of grown. So let's assume we get a paper with this sentence. In addition, the protein GRB2 can associate with GEB1. Well, we generate an entity, an object statement of a type statement. It is extracted from a certain journal. It has all this contextual information attached to it. Then in the second step, we generate the event. Again, it's just the individual. and it has certain structure and it's connected to other entities, biological entities, like a protein GRB2 in mouse or even the other protein GEB1 in mouse. It's of certain type, the type is binding, and well, there is a bit of ontological reasoning going on in the back, so it's we know binding is a subclass of event. And as I said, we can actually find many of these statements, and that's part of the whole challenge, how to manage this redundancy. Well, first of all, we represent this redundancy, so we create another statement which is based on the finding GRB2 does not interact directly with GRB1. So, let alone all the other contextual information, the crucial difference here is that the truth value is opposite. So this one says this interaction does not hold at all. And now the challenge with using, now half of the challenge of knowledge assembly, or as I call it also knowledge reconciliation, comes to ensuring that we always point into to the entities, we preserve identity of entities that appear in our model. So in this case, it's extremely important to determine that those two statements actually speak and point to the same entity there, which is a GRB2 binding with GRB1. And this is easier said than done, because actually deciding whether two entities are identical or not requires quite a lot of reasoning. You have to go through the structure, you have to see whether the identifiers and the whole structure are the same, and then you can decide, well, this is the same event. Now, given this graph, the major question is now, knowing all these things about evidence, what can we really say about the truth of events? Because at the end, we want to have domain knowledge, not just the knowledge extracted from papers. And here's where the probabilistic part of the whole approach is involved. So basically, I don't know where is this? We employ probabilistic models, a probabilistic reasoning framework that allows us to aggregate support for particular interactions and particular events. So in this simple example, I mean, the intuition is very straightforward. You have true and false events with certain probabilities, and you can see once you add more to the system, this is the intuitive behavior of the system. This is how we want the system to behave. We want to increase the probability of the event being true once we get a positive statement. We get probability of the interaction being false once we receive a negative statement, and we also notice and acknowledge the appearance of inconsistency between the evidence. In fact, there is a bigger model. This is the model behind it in a simplified form. It's all about managing redundancy, as I said, or another way to put it, it's like constructing a complex voting system where it's not a really democratic one, because not every statement has the same power, the same weight to contribute, but it's about aggregating level by level those statements and understanding at the end what's the overall negative support of a certain interaction and what's the overall positive support. So then we can kind of have a big picture to what extent such events are supported, or actually the opposite, or their negation is supported by the collected evidence. All right, once we have the picture, once we have this knowledge collected, we can, as I said, consider using an expert. In our case, actually, we are very lucky to be able to use a very fancy equipment, which is a robot scientist developed in Manchester. Some of you might have heard of it. It's supposedly been quite known in the UK, in scientific world. We can, whenever we have a burning question, we have some high conflict between the evidence, or we don't have enough arguments to support certain interaction, which is important, we can run such experiment, and we can again feed the results back to the system and adapt our levels of belief in this certain event. So I'm finishing here. So just to summarize, the big mechanism technology is there for extracting such large mechanistic explanatory models and operationalizing them. So basically offering them in a form accessible to computational agents. And as I said, there are many of such of such big mechanisms, of such models in different domains. There are ecosystems, there are brains, there are economic and social systems. And the overarching goal of the project is to try to build generic technologies. Now, with the approach we are trying to pursue, so probabilistic knowledge assembly, we believe this is a sort of generic solution for scalable and flexible knowledge assembly. So knowledge assembly that tells you what we know to the extent that we are justified to believe in it. Obviously, by the use of semantic technologies, we have a very uniform knowledge representation model and data access interface, which is always good, especially that it's supposed to be a broadly collaborative process of constructing such systems. The use of declarative formalisms, both the linked data standards and also, which I didn't mention, but it was there on the slide, the use of probabilistic logic programming tools that we use for doing the whole probabilistic reasoning facilitates provenance tracking. And this process, so basically we can, given the results, given the knowledge output at the end, we are able to tell why this happened and how come we are sure that this is actually the case. So it's easier to go back and understand the premises that led us to these conclusions. And as I emphasized at the beginning, the important aspect of this is to have a system which enables you to keep this constant update and assembly loop in dynamic environments like science, where the results are being published, are being validated, sometimes confronted with each other. So the system is almost alive there under this link. And well, I'll stop here. Thank you very much.
You want to ask a question?
Super cool presentation, by the way. Yeah, I have two questions. I'll try to keep it short. The first one, how did you do the extraction of the facts out of the articles? Did you try to develop that part of the system, or is there already some stack out there that is already available for people to use?
Yes, we would need to have folks from Manchester. There is a group responsible entirely for text mining. We receive their results, and from then on, it's our job.
Is that from also from that part of the text mining, you can identify what the what are the events that the statement should be extracted into as well, or is that a manual process?
This is so they do recognize entities and they do characterize the main types of events, so the categories of types. We actually break it into richer representation and enrich this basic categorization of entities with extra data sources. Sorry?
Is that a manual process or is that automated?
No, this is all automated.
Okay.
So this is the goal of automating this whole pipeline.
Super cool. So this is like automatic knowledge acquisition as well from that.
Yeah.
Yeah. So the second question is that can this whole thing be an ETL pipeline, a generic ETL pipeline? Extraction, transformation, loading, if you're trying to aggregate multiple different sources of data, which is not necessarily publications, but just like multiple sources of data on the internet that you want to aggregate into one base.
It is in principle, but it's also more than that. It's supposed to be more than that. It shouldn't be just one directional, taking you from the unstructured data to the result. It should be all the time involve this iterative loop where you can add new input, validate certain statements, and learn also in the opposite direction. Once you have some more confidence in expert opinion, you can actually relearn the probabilistic weights to get more accurate results at the end.
Okay, really cool. Thank you.